Hey everyone, and welcome back for another deep dive. Today, we're going to be looking at some really fascinating research on a group of proteins called Adhesion G protein-coupled receptors.
Or aGPCRs.
Yeah, aGPCRs for short.
Right?
Now, you might be thinking proteins.
Yeah.
How exciting.
Right.
But trust me, this is really interesting stuff.
It is.
Especially because almost 35% of all FDA-approved drugs target a really closely related group of proteins.
Wow.
But not aGPCRs.
Yeah.
Until now.
So what makes aGPCRs so interesting is that they play this like dual role. They're like the glue that helps your cells stick together.
Okay.
But they also act like these tiny antennas.
Like sending and receiving signals.
Exactly. Sending and receiving signals within your body.
Oh, wow.
They're involved in all sorts of vital processes, like how tissues grow, how your immune system functions, even the development of your organs.
Yeah,
and when things go wrong with aGPCRs, well, that's when we see problems like cancer, neurological disorders, and a whole bunch of other health issues.
So these aGPCRs, they're clearly really important in our bodies.
Yeah.
But I'm kind of hearing there's been a bit of a roadblock in studying them.
Yeah, there has.
Like they've been holding out on us.
They have. It's true.
Sharing their secrets.
You see, aGPCRs are pretty complex. They have this long, intricate section that sticks out from the cell surface.
I'm picturing like a long, dangly arm.
Exactly.
Yeah.
And for a long time, scientists just haven't had the tools to see exactly how that arm interacts with the part of the aGPCR that's embedded in the cell membrane.
Oh, wow.
It's like trying to understand a machine with only half the instructions.
So scientists had this partial picture.
Yeah.
But couldn't quite figure out how all the pieces fit together.
That's a great way to put it.
Yeah.
But thankfully, a team of researchers at the University of Chicago decided to tackle this challenge head on.
Right.
They used a really clever combination of techniques.
Okay, so spill the tea. What do they do?
Okay, so first they use this really cool technique called cryo-electron microscopy.
Cryo-EM.
Cryo-EM, yeah.
I've heard of that.
It's basically like hitting pause on a molecular dance party.
Oh, wow.
You flash freeze these molecules, and you can get an incredibly detailed snapshot of their structure.
So they froze these aGPCRs mid-move to get a good look.
Exactly.
It's pretty ingenious.
But they didn't stop there.
Okay.
To really stabilize the aGPCR and get the whole picture.
Yeah.
They also used a specially designed antibody that acts like a molecular scaffold.
So it's holding everything in place.
It's holding everything in place.
Right.
And by combining those two techniques, they were able to capture the complete structure of an aGPCR for the very first time.
So it's like they were putting together a 3D puzzle.
Yeah.
And they finally found that missing piece.
Right. But they didn't just study any random aGPCR.
Oh.
They focused on one called letrofilin-3.
Letrofilin-3.
And it's known to play a role in brain synapse development.
Oh, wow.
And it's also been linked to ADHD and some forms of cancer.
Okay, so understanding how letrofilin-3 works could have
huge implications for all of those conditions.
That makes sense.
Yeah.
Start with one aGPCR.
Yeah.
And hopefully unlock the secrets of all the others.
Exactly.
So what did they find out about letrofilin-3?
Oh, was there a big aha moment?
There definitely was.
Okay, you've got me hooked. What did they see?
Okay, so before this study, scientists thought aGPCRs were activated in one specific way.
Like flipping a switch.
Yeah, like flipping a switch.
On or off.
It was believed that a molecule outside the cell would bind to that long, dangly arm we talked about.
Right.
And that would cause it to split apart. A process called cleavage-dependent activation.
Cleavage-dependent, yeah.
Right.
But when they looked at the cryo-EM images, surprise.
Okay, what did they see?
The images revealed that this arm could actually adopt multiple positions.
Oh, wow.
And each position creates a different point of contact with the part of the aGPCR that's embedded within the cell membrane.
So instead of a simple on-off switch, it's more like a dial with different levels.
Exactly.
Okay.
So that changes things. Yeah, it opens up the possibility that aGPCRs could be activated in a totally new way.
Okay.
Without needing to chop off a piece of that arm. And this could allow for much more subtle control over cell signaling.
Wow.
But the researchers didn't just stop at the cryo-EM images.
Okay.
They wanted to be extra sure.
Of course, scientists love to double-check their work.
They do.
So how did they confirm that these different positions actually lead to different levels of activity?
Well, they used another technique called Foerster resonance energy transfer. But let's just call it FIRST-RE for simplicity.
FRES-A.
Okay.
Fred, yeah.
So what is that? What is FIRST? Basically, they attach tiny glowing tags to the aGPCR.
Okay.
So it's like putting a little tracker on it.
Okay.
And this allowed them to actually see how the aGPCR moved and changed shape in response to different forces.
Wait, so they could actually watch this aGPCR in action?
They could.
Sending these different signals based on how it was moving.
Amazing, right?
It's like they were watching a tiny dance.
Yeah.
And each move sent a different message.
That's a great way to picture it.
Yeah.
And what they found was truly remarkable.
Okay.
They observed that each position of that long, dangly arm corresponded to a distinct level of signaling activity.
Okay.
So it confirmed their suspicion that there's this whole new way to think about how aGPCRs work.
This is big news.
It is.
But before we jump into the implications of all of this, let me just make sure I understand the key takeaway so far.
Okay.
We went from having a very limited understanding of aGPCR structure to actually seeing a detailed image of this entire molecule.
Yep.
And now we have a whole new understanding of how they might be activated.
Exactly.
Right?
You got it.
This is a major breakthrough.
It is.
In our understanding of these really critical players in our bodies.
Yeah.
And now for the really exciting part, what does this all mean for us?
Right.
But we're going to have to save that for part 2.
Yeah, stay tuned.
Okay.
And the possibilities are pretty amazing.
That's what I was thinking. Like, we've suddenly got this whole new playbook for targeting these aGPCRs with drugs.
Yeah.
So let's talk about what this could mean for treating diseases.
Well, one of the most exciting things is the potential for creating drugs that are much more precise.
Oh, okay. How so?
You see, each aGPCR has a unique shape, particularly that long arm that sticks out.
Right.
It's like a fingerprint.
So we could design drugs that fit a specific aGPCR, like a key and a lock.
Exactly.
With less chance of affecting other proteins and causing side effects.
Yeah, that's the idea.
Okay, I'm starting to see how this is a game changer.
It is.
And what about those antibodies the researchers used
Right.
to stabilize the aGPCR for imaging?
Well, think about this.
What?
What if we could engineer similar antibodies that don't just stabilize the receptor, but actually control its activity?
Oh, wow.
We could potentially create antibodies that switch a specific aGPCR on or off, or even fine-tune its activity to just the right level.
Whoa. That's like having a remote control for these cellular superheroes.
That's a great way to put it.
What kind of diseases could we potentially treat with this kind of approach?
The possibilities are really vast.
Yeah.
Because aGPCRs are involved in so many crucial functions in your body.
Right.
We could be looking at new treatments for everything from cancer to neurological disorders to immune system dysfunction.
So, for instance, how could this new understanding of aGPCRs help us fight cancer?
Right. So some aGPCRs seem to be involved in how tumors grow and spread.
Right. You mentioned that earlier.
Yeah.
That there's a link between certain aGPCRs and cancer.
Exactly. So imagine if we could design a drug that specifically targets those aGPCRs, blocking their activity.
Okay.
It could potentially stop a cancer in its tracks.
Wow.
Or even shrink existing tumors.
That would be incredible.
It would.
And what about neurological disorders like ADHD?
Yeah.
Which you mentioned earlier is linked to letrozole 3.
Right. That's another area where this research holds a lot of promise.
Okay.
If we can figure out how to fine-tune the activity of letrozole 3, we might be able to alleviate some of the symptoms of ADHD.
So we could potentially address the root cause of the problem.
Yeah.
Not just manage the symptoms.
That's the hope.
Wow.
Of course, there's still a lot of research to be done.
Right.
But this new understanding of aGPCRs opens up so many avenues to explore.
It's like we've gone from fumbling in the dark to having this amazing map that shows us exactly how these cellular processes work. And now we can start to figure out how to intervene in a much more targeted way.
Exactly.
That's pretty cool.
It is.
And the amazing thing is, this is just the beginning.
Right.
There are 33 different aGPCRs.
In humans.
Yeah.
Yeah.
And we've only just begun to uncover their secrets.
Exactly. Each one could hold the key to treating a different disease.
That's such an exciting thought.
It is.
This one study has opened up a whole universe of possibilities.
Yeah, and for our listeners who want to dive even deeper into this research, we'll be sure to include links to the original study in Nature Communications and the University of Chicago press release in the show notes.
Perfect.
It's definitely worth a read.
Yeah.
This is the kind of research that has the potential to revolutionize medicine.
Yeah, absolutely.
As we know it.
This has been an amazing journey into the world of aGPCRs.
It has.
But before we wrap things up, I'm curious, with all these different aGPCRs out there, what other secrets do you think they might be hiding?
Ooh, that's a good question.
And how could those secrets help us live healthier lives?
Right.
Let's ponder that as we move into part 3 of this deep dive. It really is amazing to think that each of those 33 aGPCRs could be like a doorway to these new treatments for different diseases.
Yeah.
Talk about job security for researchers like you.
Yeah, that's right.
So as we kind of wrap up our deep dive into the world of aGPCRs, what's like the one thing you really want our listener to take away from today?
Well, I hope they come away with a sense of awe for the incredible complexity of their own bodies.
Yeah.
You know, these aGPCRs are doing so much behind the scenes to keep us healthy, and we're only just beginning to understand how they work.
And to think for years, these proteins have been kind of overlooked.
I know.
Right?
It's true.
But this research shows just how much potential they hold for treating a whole range of diseases.
It really highlights how important basic research is.
Yeah.
Sometimes the biggest breakthroughs come from exploring the things that we don't fully understand yet.
I like that saying.
Yeah.
The more you know, the more you realize you don't know.
Exactly.
This research has answered some big questions about aGPCRs.
It has.
But it's also opened up this whole new set of questions to explore.
Right. And who knows what amazing discoveries are waiting for us down the road.
It's exciting to think about, isn't it?
It is.
This one study has completely changed the way we think about aGPCRs.
Yeah.
And it's opened up this whole new chapter in the search for treatments and cures.
Right.
It makes you wonder what other hidden heroes are lurking within our cells, just waiting to be discovered.
That's a great way to put it. I think that sense of wonder is what drives so many of us in science.
Yeah.
There's always something new to learn, always another puzzle to solve.
Right.
And, you know, those discoveries could one day transform the way we approach healthcare.
Well, on that note, I think it's time for us to surface from this deep dive.
All right.
Thanks so much for taking us on this incredible journey into the world of aGPCRs.
My pleasure.
And to all of our listeners out there, thanks so much for joining us on the deep dive.
Yeah.
We'll be back soon with another fascinating exploration of cutting-edge research and its impact on your world.
Can't wait.
Until then, stay curious.